Okay, don't worry. I have not forgotten about the AP Bio crash courses. We are doing them. Okay? Totally didn't forget. Hello everybody, I'm Kara, and today we are going to be doing Unit 5, Heredity, which is going to be very epic. As you can see, we're talking about meiosis and Mendel and all that good stuff. So why don't we just get into it? So in the last unit, Unit 4, we talked about mitosis, right? And mitosis makes identical daughter cells, right? If you want to grow, right, you want to just duplicate the exact same cell, use mitosis. But you know that, like, you're not the exact same person as your parents. It would be very, very creepy if you look exactly like both your mom and your dad at the exact same time. Now, that would be really weird. So somehow we had to combine both your mom and your dad, right? So we know that your mom has, like, cells, body cells with 46 chromosomes. You know your dad has a body cell with 46 chromosomes. And you know that to make a kid, to make a zygote, right, you basically need an egg cell from your mom, and you need a sperm cell from your dad. They need to join up like cool kids, and they have to make a zygote, you. But you have 46 chromosomes. So if you have 46 in the sperm and 46 in the egg and they come together and you make 46... Wait, no, that's not how addition works. 46 plus 46 is not equal to 46. So clearly something is wrong here. Somehow we need to make it so that the egg and the sperm have 23 chromosomes. Otherwise you're going to literally have 92 chromosomes, which is not right. Also, just another thing to remember, make sure that you remember that this is called fertilization, right? Basically you get some stuff from your mom in the egg, you get some stuff from your dad in the sperm, and you make yourself a zygote. Okay, so basically the way you make these egg and sperm cells, right, they're not normal body cells. We need a new type of cell division that actually has your chromosomes, right? You go from 46 to 23 somehow. And that is called meiosis. Alright, so why don't we talk about how meiosis works? And I really think the best way to remember how any process works, right, is to just think of the endpoints, right? What do you start with, what do you end with, and then it's easy to, like, figure out the steps in between if you know the starting and ending point. So, for example, right, mitosis, you have like 46 chromosomes. Let's just say we have 6 chromosomes because I am too lazy to draw 46 chromosomes. Then basically this guy divides up into 2 cells with the exact same chromosomes, except now they're no longer duplicated. Alright, so this is mitosis. You make 2 identical daughter cells. Now meiosis is a bit more complicated because now you have to keep track of which ones you got from your mom and which ones you got from your dad. So let's say you have like 3 chromosomes from your dad, right? Half of them from your dad, and the other half are from your mom. And basically you know that your mom and dad have like the same types of chromosomes, right? Each of your mom and your dad both give you a chromosome 1, each of them give you a chromosome 2, each one give you a chromosome 3, all the way up till they both give you a chromosome 23, which are just your sex chromosomes. But basically, we will number these, right? So your mom gave you a complete set, right? 1, 2, 3, and your dad also gave you a complete set, right? 1, 2, 3. And basically what meiosis does is it randomly puts like all these chromosomes into different cells. And you can see, right? Like mitosis basically keeps the number of chromosomes the same with one division, right? So we want to divide again in order to have the chromosome. So if you multiply by 2 every time you divide, you go 2, and then you can do another 2. Each of these guys split into 2, and you're left with 4 daughter cells, each with half as many chromosomes. And each of these things are randomly distributed. Let's just chuck them in there. And each of these daughter cells is basically a gamete or like an egg or sperm cell. Now also keep in mind that each of these has to have a complete set of chromosomes, right? You can't just give your kid like only a 1 and a 2. That'd be sad, right? Your kid's losing out on a 3. Come on. You gotta give them all of the types, right? So basically, like, each of these has a full set. So let's say it's like 1, 1, 3, I don't know. So you can see, right, we started with 2 1s, right? We started with 2 2s, 2 blue 3s, and you can see they're all in the cell somewhere. Alright, so now we know the start and end points of meiosis, right? You start with a cell, a full 46 chromosome cell with duplicated chromatids, right? And you end up with 4 separate cells, each with half as many chromosomes, and each chromosome is not duplicated anymore. And your mom and dad's characteristics are randomly chucked in there. And so what's important to note about the randomness, this is one part of sexual reproduction, right? The fact that we have 2 people coming together is the fact that we randomly put these chromosomes in our gametes, right? So that's one way we get genetic diversity. Okay, so why don't we actually talk about how we get from A to B? How do we get from the start point to the end point? So basically the way I like to remember it, right, if you have 4 daughter cells, you need 2 divisions, right? Each division divides it into 2, so if you need to get to 4, you have to divide it twice, right? And essentially, each one of these divisions is just a mitosis, right? So meiosis is basically 2 mitosises. Alright, so let's just do mitosis once, right? So you know that the first stage of mitosis is prophase. So in meiosis, this is called prophase 1. And basically, in mitosis, this stage is just setting up for division, right? Your chromosomes condense, stuff gets connected to the centrioles, all that good stuff, your nucleus membrane dissolves, all that stuff. But in meiosis, you want to generate a bit of variation, so in prophase, they also prepare, except they do a little bit more fancy stuff called crossing over. Crossing over, and basically this is all you have to know about prophase 1. The one important thing, if you have to know anything about meiosis prophase 1, is crossing over happens. So basically, these 2 guys pair up, these 2 guys pair up, these 2 guys pair up. If they have the same number, right? If they're both chromosome 3s, if they're both chromosome 2s, they pair up. And basically crossing over is literally what it means, right? You see these guys are crossing over, right? They basically switch parts, right? They basically exchange little parts of their chromosome, so now there's a bit of variation, right? It's not entirely your dad's chromosome, it's not entirely your mom's chromosome. So that is another way that meiosis generates genetic diversity, right? It randomly switches parts between your mom and dad's chromosomes. And basically all the stuff that happens in mitosis prophase also happens in meiosis prophase. Also, just a little bit of terminology, right? Basically the parts where they cross, like where they switch over is called chiasmata, right? The X's that they make, right? Like this X right there is called a chiasmata. And basically the thing that holds them together is called the synaptonemal complex. None of this is that important. I doubt you'll actually have to know this stuff, but I'm just saying it in case it comes up. Just to refresh your memory if you got know it already. Okay, so why don't we move on to metaphase now? So we're still on the first mitotic division, so it's metaphase 1, right? And basically, in prophase, all this stuff got connected to the centrioles, right? All that good stuff. And basically, metaphase is the exact same thing as mitosis metaphase, right? Basically, the things that are already paired up, right? These guys are paired up, all of them are paired up. They basically align at the center. So you got this pair at the center, you got this pair at the center, you got this pair all aligned at the center. Metaphase, I just remember as middle, right? M for middle, align at the center. Okay, now the next phase, what do you think it is? Look at mitosis, and it is anaphase, PMAT, very cool. So we're still on anaphase 1 now. And once again, this is exactly the same as mitosis, except this time, instead of mitosis, where it splits off like individual of these chromatids, it just splits off like these entire chromosomes. So basically, your mom's chromosome 1 will split off from your dad's chromosome 1, and now they'll go separate ways. So what it looks like after anaphase, right? You basically snatch your one of these, right? Let's just say your mom's went this way, your mom's 2 went this way, your dad's 1 and 3 went this way as well. And then same on the other side. Okay, so now you can basically see, right? We have the number of chromosomes. Now instead of having one copy from your dad, one from your mom, you only have half as many chromosomes. Each of those chromosomes is still duplicated, right? But now you have half as many. You only have one set of 1, 2, 3. In a normal cell, you have 2 sets of 1, 2, 3. In this one, you only have one set of 1, 2, 3. And as you can see, these chromosomes separate randomly, so there's one place where genetic diversity is generated. And then telophase is basically just where it extends, it starts blobbing like a cool kid, and eventually cytokinesis splits it like in normal mitosis. Okay, so now let's move on to the second mitosis. Okay, so after telophase and cytokinesis, you're left with 2 cells, right? And now they both have half as many chromosomes, right? You might have started with 46, your diploid number, but now we're at half as many, right? Haploid, right? Diploid is 2, haploid is half. Very cool. So now we do the second mitosis, right? Now it's on the second version, so we call it prophase 2. And basically what's cool about the second mitosis that happens, right? The second division, is it's exactly like mitosis, okay? Like literally it's exactly the same. So prophase is just break down the nuclear envelope, condense your chromosome, detach stuff, very cool. Then metaphase, you align at the center, right? Very cool. And then after this, and after we're done with metaphase, we go to anaphase. And just like in mitosis, the actual chromatids, right? The identical chromatids on this one chromosome that may have been changed by crossing over, now split apart. So at the end of anaphase, these guys have been drawn to other sides. So this is how it looks after anaphase, and then we finally go into telophase. So now it starts blobbing, and then eventually these guys split apart by cytokinesis, and you are finally left with what we said we would end up with. 4 epic daughter cells. Epic! We are finally done with meiosis. Now, keep in mind that these are not purely your dad's or purely your mom's, right? Remembering crossing over, some parts of them might have flipped over, so that might have changed a little bit, but this is what you basically end up with. So you started with a haploid, so you started with a diploid 46, and you ended up with a haploid 23. Now, if you don't want to memorize the entire sequence, although I would recommend you do because it makes it a lot easier to understand, basically just remember you start with a completely normal body cell, and you end up with 4 daughter cells, each with half as many chromosomes and non-duplicated chromatids. And basically the way I just like to remember meiosis without having to think about it at all, just think of it as 2 mitoses, right? Like the first time you split up homologous chromosomes, right? Your ones from your dad and your mom are called homologous because they both have the same number. And then the second time you split up the chromatids, right? Basically they're exactly the same, you just split them up like completely normal mitosis. Alright, so now that we know how meiosis works and how it generates genetic diversity, now we can start talking about how that genetic diversity, we could actually use math to solve that. And basically the dude who made this all possible is Mendel, okay? Now we're going to talk about Mendelian heredity. So we basically already know that you basically get 1 set from your mom, 1 set from your dad, okay? And basically what Mendel said is that each trait in your body is controlled by these 1 set from your dad, 1 set from your mom. And he basically called different versions of the genes alleles, okay? So alleles are literally just versions of a gene. You don't have to think about it in any other way, just know that they're versions of a gene. And basically these genes, which are part of your genotype, right? Your genetic code, right? It basically started with gene, literally, genetic code, influences your phenotype, right? Based on what proteins your body makes, based on what genetic code you have, your appearance is affected, right? How you behave, what color hair I have, it's all phenotype, right? What it looks like on the outside. Alright, so this is the basic terminology. Now let's just talk about the most common form of inheritance that Mendel studied, right? Basically that's dominant and recessive alleles. So you got your dominant alleles, right? Basically, let's say you have a gene R, Mendel would represent it with a capital R, and this is basically the determining allele, right? If you have this allele, it will show up, right? And then recessive is the little lowercase thing, and it only shows up if there's no dominant. Only shows without dominant. Alright, so why don't we just take, for example, this is not a real example, but let's just say the dominant allele R gives you black hair, right? And the little r, the recessive allele, will give you brown hair, right? Now, basically, if you have RR, right? Let's look at our definitions. It's the determining factor, right? So if you only have the dominant, you're for sure going to get the dominant, right? Like literally the dominant one is determining, if it's there, it's for sure going to determine what you look like. So this means you have black hair. And because you have two dominant alleles, it's called homozygous, right? Homo means the same, so that's how you remember that. Homozygous means two of the same thing, and then dominant means you have two of the same dominant allele. Okay, now it's a bit more trippy when you have R and little r, right? Because then you have a black version and a brown version. So which one's going to show? Now obviously the dominant one is going to show by our definition. So this guy has black hair. And this is called heterozygous, right? Because hetero means different. And then finally, let's just say we did RR, right? Now in this case, it only shows without the dominant, right? But the dominant is not there. So essentially, this guy is going to have brown hair. And what do you think it's called, right? If it's homozygous dominant, what is this one going to be called? That's right, it's homozygous recessive. Alright, so now we know basically how alleles work. Basically, one R is from your mom, one R is from your dad, and when they combine, we could basically predict what's going to show up based on whether it's dominant or recessive. Okay, but because biology is not biology without a little bit of math, I don't know whether that's a thing, but Mendel decided to add in a little bit of math there, so you gotta know the math. This is it's pretty simple math, but you gotta know it, okay? And basically the question we're trying to answer, right, is let's say we have your mom has two dominant alleles and your dad have a dominant and a recessive allele. What are you going to show up, right? What probability either they have black hair, what's the probability you have brown hair, right? Now you can basically logic it out, right? Basically your mom's randomly going to give you one of her two alleles, but they're basically the same thing, right? So she's for sure going to give you an R, right? And your dad has a 1/2 chance of giving you a big R and a 1/2 chance of giving you a small R, right? So essentially just by looking at it, you can basically say that there's 1/2 chance of getting a big R, and a 1/2 chance of getting a little R. And that means that no matter what, you're going to have black hair, right? But just looking at it is boring, okay? We want to make beautiful diagrams. We want something nice to look at, okay? So now we're going to make something called a Punnett square, okay? And basically, these guys are kind of epic if you get more genes or something, or if you're want to just do it quickly. You basically just put each allele one place, right? You have R, you have R, little r, and now you just write whichever one it corresponds to, right? So R. Okay, so right here you can see that 2 of the 4 squares are filled with this guy, so there's a 1/2 chance of this. 2 of the 4 are filled with this, so there's a 1/2 chance of that. And basically that means that no matter what, you're going to have black hair. So now the more interesting one is if you have 2 heterozygous parents come together. Now this thing would change to a little r, and this would become 2 little r's, right? Because it comes from here, comes from there. Okay, so now only one of the 4 squares is homozygous recessive. Only one of the 4 squares is homozygous dominant, and 2 of the 4 squares are heterozygous. So that means there's a 1/2 chance of heterozygous and 1/4 chance of both the homozygotes, right? But all 3 of these result in black hair, so you have a 3/4 chance of black and a 1/4 chance of brown. Alright, so that's basically how you do it for one gene, right? Basically, this one gene has 2 versions. You have one for your mom, one for your dad, and basically this is how you would calculate what the probability of getting a certain pair from both your mom and your dad is. Now what happens if we want to talk about multiple genes, right? Let's say there's another one, Q, right, that represents, blue eyes. I don't know why blue eyes would be dominant, but we're just going to go with it. And Q is black eyes, let's say. Even though probably that's going to be dark brown, god dang it. And then basically we want to see, given that your mom and your dad both are heterozygous for both blue eyes and for black hair, what is the probability you also get blue eyes and black hair? So why don't we do this logically, right? So your mom is going to be RRQQ, and your dad is going to be RRQQ. So basically the way they teach you in school, right, you just draw a really big Punnett square, right? And basically you just do it this way, okay? So basically what they tell you to do is they're what possible pairs could you get from your mom and your dad, right? You get an R from your mom, an R from your dad. You could get a little R from your mom, a big R from your dad. You could get a big R from your dad, mom, a little R from your dad, and you could get both little R's from both of them, right? So let's just write those in. And then the same thing for your Q's, right? You could get a Q from both your mom and dad. You could get a Q from your mom, little Q from your dad. You could get a little Q from your mom, a big Q from your dad, and you could get 2 little Q's. and then you just fill in this whole thing. It's kind of nasty. Now, my approach is a bit simpler, right? Basically, it's asking how many ways are there to get black hair and blue eyes. So let's just see what the probability, ignoring the blue eyes, of getting black hair. We already know that's 3/4, right? Okay, and then if we just look at only blue eyes, there's a 3/4 probability of just getting blue eyes if we ignore the black hair. So now the probability of both happening, this is a normal, like, easy probability, right? If it's and, you multiply them. If it's or, you add them. You basically just multiply these, right? Because you want black hair and you want blue eyes. So your answer is 9/16 without actually having to do a really big Punnett square. You can just do 2 mini Punnett squares and then solve for the actual answer. Another thing you might have to deal with is like pedigrees, right? Basically, they represent family relationships, right? You might have a circle to represent a woman, a square to represent a man, and then if you want to represent they had a baby together, you just put a cross there, and then you do that, and very cool. Now they have a bunch of kids. Epic. So if they had a girl, a girl. If they had a girl, another girl. And then if they had a boy, you do that. Okay. So let's say we were talking about Tay-Sachs disease, right? And that's basically going to be a recessive disease, right? If you have 2 of the recessive allele, you're going to have Tay-Sachs. Then basically, if we wanted to say that the woman had Tay-Sachs, then we would put her in a darker color, right? Shade her in. And then a question we could ask is like, we know that our mom is little r, and let's say we're given that the dad is big R, then what are the probabilities that our kids have little r, right? And basically that's just 1/2, right? Because there's guaranteed to get a little r from mom, and they could only get it if they get a little r from dad, so it's a 1/2. So essentially, about 1/2 of the children should have Tay-Sachs, so that seems legit. Okay. So this is called a pedigree. Just know, like, the symbols, right? Basically, if you have a circle, it's a woman. If it's a square, it's a dad. And if you have a line connecting them, they're related. Okay. So now I basically touched on this indirectly, right? But the 2 laws that you have to take away from all the stuff we've been talking about are law of segregation and law of independent assortment. So basically, any gene that follows Mendel's rules, right? Basically, you have like 2 versions, right? And they both come together from your mom and your dad. They follow these 2 laws. So segregation basically means that if your mom has an R and an R, they're going to segregate in her gametes, right? Each gamete is only going to have one of these alleles, right? Segregation, split them apart. Now, law of independent assortment is if you have like RRQQ, how RR is split up among the gametes is irrelevant of this guy, right? Like, this guy's going to split up completely separately. RR is going to split up completely separately. Law of independent assortment just means that 2 different letters, right, 2 different genes are not affected by each other at all. Now, of course, the problem is that these laws have their exceptions. So now, the last thing we're going to talk about today is the exceptions to all these rules. So the first exception I want to talk about is incomplete dominance, right? And that basically means that, like, if you have an R and an R, this is actually going to be different from big R, right? So let's say R is like a protein, right? That, I don't know, like, metabolizes lipids for, like, Tay-Sachs or something, right? Then essentially what incomplete dominance means is that if you have the big R and the little r, it'll actually produce less of the protein than big R, right? Because only the big R actually produces the protein, and if you have little r, it's actually going to not produce any protein whatsoever. So no protein, a little protein, and a lot of protein. So that's basically incomplete dominance, basically when heterozygous is actually not as much as the, like, homozygous dominance. It's incomplete, right? Like, the big R by itself is not enough to completely overrule the recessive allele. Now, the next one is codominance, which is really, really similar. I honestly keep getting confused between the 2, but basically my understanding is that codominance means that both alleles code for slightly different things. So essentially, I think a really good example is the blood groups, right? Basically, your blood has, like, things on the surface, right? And there's 2 types of them, right? And basically, an example is, like, if you have RA and you have RB, right? Then you're going to have both A and B, like, molecules on the surface. Now, if you have RA, you're only going to have A, right? And if you have RB, then you're going to have only B. So both of these are dominant, right? They both result in a phenotype, right? But, like, and both of them could be together, but neither is, like, completely dominant over the other one. Now, your ABO blood group, which is a much more familiar blood group, is actually controlled by multiple alleles. So that is another exception to the rule. So instead of just having a dominant and a recessive, or just 2 versions of the allele, you might have multiple versions. And in the ABO blood group, there's actually 3 alleles. There's an IA, there's an IB, and an I, little I. So if you're AB, you have an A and a B. If you have IA, little I, or IA, then this is going to be an A-type blood group. Now, if you have IB, little I, or IB, then you're going to have a type B. And finally, if you have 2 little i's, you're basically going to have 0, right? You don't have any of the other things. You're going to have an O because you don't have an A or a B. There's something called pleiotropy, which basically means that one gene affects multiple traits, right? So that's what pleiotropy is, and then polygenic inheritance basically is the opposite, right? Multiple genes affect the same thing. And basically the way I remember the difference between the two is like pleiotropy, right? Like, I don't know, it kind of sounds like it's affecting multiple things, but polygenic means multiple genes, right? So essentially, pleiotropy had nothing to do with multiple genes, so it must be one gene doing multiple things, but polygenic has the gene literally in it, so it's multiple genes affecting the same thing. Okay, so now one of the biggest exceptions that you're going to have to know are linked genes, right? Basically, if multiple genes are on the same chromosome, right, the only way like let's say there's a gene A right here and there's a gene B right there, the only way that these two things could split apart is if during crossing over, that very exact spot switches over with the other chromosome. So basically, the farther apart A and B are, the more likely they are to be completely independent of each other, but they're still linked as long as they're on the same chromosome. And when these are on sex chromosomes, right, which are your X and Y chromosomes, this is called a sex-linked gene. Now, what's interesting about sex-linked genes is that they're completely different, right? Because a boy and a girl like get different X and Y chromosomes, right? So essentially, the way we represent genes on an X chromosome, we do XA and X little a, and let's just do the example of hemophilia, which is an X-linked recessive disorder. So if you have two XAs, right, then you're going to have hemophilia, right? So remember, girls have XX, right, and boys have XY. So essentially, whenever you like have a girl and a boy together, you basically have two X's and one X, one Y. So essentially, that means that the girl needs two A's in order to be sick, but if the boy only has one A, right, he's already sick, right, because there's no dominant allele to mask the recessive allele. So that's why boys are a lot more affected by X-linked diseases, right? So in hemophilia, let's just do an example. So let's say that your mom is a carrier, right? She has one version of the hemophilia gene, right? And let's say your dad is a hemophiliac, right? Then that basically means he must have the recessive allele. So now, if we do a Punnett square real quick, we basically get an XA, 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 XAY, and XAY. Okay. So basically, if they have a daughter, right? These two are daughters, right? There's a 1/2 chance that they have hemophiliac. There's a 1/2 chance that they're a carrier, right? They have the disease gene, but they're not actually diseased, right? If they have a son, then there's still a 1/2 chance, right? Because only this guy's gonna have it, this guy is not. Okay, what happens if we make this XA? Okay, then this would become XA little a. So if the mom doesn't have a recessive allele, it's impossible for either the daughter or the son to get the disease, right? Because no matter what, your dad's gonna give the son the Y, so there's no way the sons could get it, and your mom's always gonna give your daughter at least one of the dominant allele, so they're protected. Okay, very nice. Alright, so now the last important thing you gotta know about linked genes is recombination frequency, okay? And basically this is what happens if you cross like RRQQ with little r, little q, and you basically find out how many of them do not look like either of the parents, right? Because basically we want to see how many times like this big Q splits up from this big R, right? Now basically for this cross there are 4 types of offspring, right? There's RRQQ, there is RRQQ, there is little r, QQ, and little r, q. Okay. So essentially what recombinant frequency measures is how many of the offsprings are different from both of the parents, right? So this parent is black hair and blue eyes, this parent is brown hair and black eyes, right? So essentially these two are the only offspring that are going to be different from both parents, right? So essentially if we're given that the percentages are 40%, 17%, 18%, and like, I don't know, what's the last thing? Okay, 25%. Then essentially our recombination frequency is just the sum of these two percentages, right? So our answer is just 35%. Okay, cool. But that is it. Alright, thank you guys so much for watching. I hope it was helpful. As always, if you enjoyed the video, leave a like and subscribe for more. Thank you guys for watching again, and see you guys next time.